Hello, students. This is a recording for Biology 1407, the first chapter in the unit on plant evolution and structure, Chapter 25, Seedless Plants from the Biology OpenStax. All right, so first we need to identify what a plant is. These are multicellular photosynthetic organisms for the most part. There are some non-photosynthetic plants, like parasites. But generally, photosynthetic multicellular organisms that live primarily on land as terrestrial. And there's an exception there because there are quite a few aquatic plants that have derived or developed from terrestrial plants, as opposed to algae, which are generally aquatic organisms. So the key distinction is multicellular versus colonial. Most algae are colonial, as well as many single-celled varieties, whereas all true plants are multicellular organisms that are non-reducible. You can't break them down into their individual cells and expect them to continue functioning. So a little bit about what a plant is. And they are evolutionarily related to one of the four groups of algae, green algae. Colloquially, you can divide algae into four groups based on their pigmentation, their photosynthetic pigments, and a bit of their structure. That would be golden algae, brown algae, red algae, and green algae. And those differences are partially due to the photopigments they use. All right, so terrestrial plants. Plants exist as these terrestrial organisms. The first question is, why are they on land? And there are a couple of theoretical answers to that are partially supported by research. One is a competitive question. As the marine environments and freshwater environments became increasingly full ecologically of organisms competing with each other, land, it was an open new habitat that, if it could be exploited, would free the first colonizers onto land from competitive pressure in an aquatic setting. So one is ecological release, the release from competitive pressure. Second is increased access to resources. Photosynthetic organisms, three resources that are needed for the synthesis of carbohydrates are, of course, a power source, sunlight, carbon dioxide as the substance to be reduced, and a source of electrons, which, of course, in photosynthesis is going to be water. If you move on to land, the amount of radiative energy, sunlight, is increased substantially. Water blocks quite a lot of that. And CO2 pressure in the atmosphere is quite a bit higher than it is in aquatic environments. So access to CO2 and light is expanded if those plants can secure access to water. That's going to be the limit of living on water. So terrestrial plants had to, A, avoid drying out, as your PowerPoint shows right here. I think, yeah, I'm pointing at the right thing. They had to avoid drying out, and they had to develop mechanisms that allowed them outside of water to, support themselves, capture sunlight, and also dispersal of reproductive cells. This is a very important thing. So we divide terrestrial plants into two groups: the seedless plants, which are restricted, hampered in reproduction on land, and then the seed plants, which has effectively solved the reproduction on land problem most thoroughly. I mean, these things succeed. Seedless plants succeed in reproducing on land, but there are difficulties that constrain where they live. All right. You want to become quite familiar. In fact, you want to commit this to memory, basically. This is in a box form, kind of in a chart form, a phylogenetic tree of the photosynthetic organisms that we're interested in. So streptophytes is a large phylogenetic group that includes all of the green plants, some of their algal ancestors, as well as all the true terrestrial plants. So under streptophytes, we have the charophytes, which is a particular group of algae that we'll look at in just a moment. and then a big group called the embryophytes. And phyte is the Greek root for plant. So when you see this word phyte or phyto or phyta, it has to do with plants inevitably. So embryophytes are a particular group of photosynthetic organisms that maintain a developing new generation of diploid organism attached to its predecessor. And that's, you know, kind of like the embryonic relationship between an animal and its offspring. So that is a unique characteristic of these things called land plants that the other green algae, including the ancestral group Charophytes, don't share with terrestrial plants. So this is the novel feature, the production of an embryo. There are some other characteristics as well. And then we take those embryophytes and we divide them up into non-vascular and vascular plants. So non-vascular plants have no dedicated specialized tissues for nutrient distribution in the organism, whereas vascular plants have a more sophisticated, dedicated set of tissues that are specific. Their specific function is to distribute metabolites throughout the organism. So they carry out the same functions that a circulatory system does in an animal. In the vascular plants, the non-vascular plants include the seedless plants, bryophytes. This is collectively what, this is the largest group name for what we normally refer to as mosses right here. But to be specific, bryophytes includes the liverworts, the hornworts, and what most people are familiar with, the mosses. But these are all related groups of organisms that have various levels of adaptations, various specializations that make them able to live and function on land, but also limitations. Again, they produce no seeds, which is a reproductive limitation, and they don't have vascular tissue, which limits their size and complexity. Then we have the evolution of vascular tissues in plants, so that gives us the vascular plants, and then we can divide those into a seedless group, which share with the bryophytes some limitations on their ability to reproduce, and then the seed plants that have effectively solved the terrestrial water-free reproductive question. So seedless plants versus seed plants. And then when we look at those seedless plants, the vascular seedless plants, they're vascular, so they're going to be bigger, more substantial. Vascular tissue allows them to be large. And so we have the lycophytes, which as we see right here are the club mosses, the quillworts, and the spike mosses. These are colloquially referred to as mosses and these things as worts right here because they share a lot of structural features with these organisms, and that's because of a evolutionary descent. These organisms are derived or evolved from various members of this group right here, from the bryophytes right here. So they share structural characters with them, but they are in fact vascular seedless plants. And then a more distinct group, the one that you're most familiar with individually, is going to be the ferns right here, collectively referred to as the pterophytes, P-T-E-R-O. This is another one of those Greek letters with two consonants at the beginning. And in these Greek pronunciations, we pronounce this second consonant. So you'll remember Cnidaria, phylum Cnidaria, the jellyfish and all of those guys. It is C-N, but we only pronounce the N. So that's the way you say this word. It's pterophyte. Mimic saying a p, but don't do it, and then you've said the word right. Anyway, whisk ferns, which is an interesting group of organisms. Horsetails, equisetum. Equus means horse, and setum is, I believe, means tail, but anyway, the horsetails. And then the ferns. Most people are familiar with ferns. And then derived from one of these groups right here are the seed plants. And again, the significance of the seed is that it's an innovation in reproduction and distribution of the young that reduces the plant's requirement or dependency on water. And those are the gymnosperms, which are non-flowering seed plants. That's pines, junipers, and all of their relations. Cypress and many other groups of plants, including cycads. And then the angiosperms, more commonly referred to as flowering plants. So that's anything that produces a flower, whether it's showy or non-showy. There's lots of green, small, non-smelly flowers, but they're still flowering plants. And so that includes most of the trees and most of the plants that you're familiar with, certainly all the grasses and anything that has an obvious flower on it. All right. So what are some characteristics that are retained by everybody in this group right here, the streptophytes and certainly the embryophytes? One is this life cycle where there's an alternation of generations, all right? So there's two other types of life cycle that you've learned before, all right? Haplonic life cycles and diplontic life cycles. And what this means is that the dominant form of the organism, the one where there's a multicellular form, is going to be haploid. And diplontic means that the multicellular form of the organism is going to be diploid. So a lot of fungi are haplontic. So when you look at a mushroom, when you look at a large fungal structure, you know, like a bracket fungus sticking off of a tree or a toadstool popping up out of the ground, or if you're thinking of, what are those called? Truffles, you know, which is a big multicellular fungus. All of those multicellular forms almost exclusively are going to be haploid. All of the cells are haploid. They still go through a sexual reproduction mechanism. So those haploid organisms produce gametes, eggs and sperm, and those gametes are fused together to make a diploid cell, which then goes through meiosis to make a fresh set of haploid cells. And that's the life cycle right there. Diplontic organisms also have a sexual life cycle. The gametes fuse together, an egg and a sperm, to make a diploid cell, but instead of immediately going through meiosis like haplontic organisms do, the diploid cells divide via mitosis to make a great big multicellular thing, the person, the animal. And all animals are diploid organisms, and most are sexually reproducing as well. So they produce haploid gametes, sperm and eggs, but that's the only cell that's going to be haploid in these organisms. Plants, on the other hand, true plants, have multicellular haploid stages, that's going to be the gametophyte, and multicellular sporophyte stages, that's going to be the sporophyte right here. And these names also describe what they produce. So a gametophyte ultimately produces gametes. It itself is haploid, and so it produces gametes via mitosis. A sporophyte is a diploid organism, but it produces spores. And the spores are haploid, so it's the sporophyte that goes through meiosis, reduction division, that we learned in 1406, and produces these haploid spores which germinate and become gametophytes. And so that's the alternating generation mechanism of growth. In 1406, certainly my students would have been exposed to this, and hopefully you were in your biology 1406 course. Now, an interesting thing about these two alternating generations right here, so all plants go back and forth between the two generations, is in the more primitive plants, which is to say these guys over here, all right, in the more primitive plants, the gametophyte is the thing that you're most likely to see. It's the bigger multicellular version of the organism. There is a multicellular sporophyte, but it's going to be smaller and more cryptic. As we look at the more sophisticated, more derived, they haven't gotten better necessarily, although they have gotten more complex and better able to live on land. The derived plants right here, we'll see increasingly that the sporophyte generation is the big thing that you see, and the gametophyte becomes somewhat less evident, even much more hard to find, and then finally extremely cryptic in the seed plants right here. They still make gametophytes, but they're hidden. You have to actually look for them in a dissection mechanism. All right, so a little bit about plants. So as plants evolved, to become increasingly well adapted to living on land, the gametophyte became reduced and reduced. The sporophyte became more and more dominant over evolutionary time. All right, so here's an illustration of that alternation of generations procedure. So what's going on right here in this drawing, 2N means diploid, so that means 2 copies of each chromosome inside a cell. That's the diploid state. And N means haploid, 1N. There's an implied 1 right here in algebra. It just means they have 1 copy of each chromosome. So when we say alternation of generations, we get this haploid cell. It's a spore. A spore is always haploid. Spore, haploid, period. That spore can germinate, and divide via mitosis. So germinating is what we talk about when we say plants. When the when a seed is germinating as well. Same idea. So what happens is a key cell in there starts dividing via mitosis, and we produce a great big multicellular organism with thousands or millions or billions or trillions of cells, depending on the details. That is the gametophyte. And so this is a plant that at some point can make specialized cells that can, find and attach to their counterpart. That's what we mean by a gamete. And of course, that's going to be a sperm cell or an egg cell. And gametes, they have one purpose, and that is to, join together with their opposites. So a sperm cell is going to fuse to an egg cell or vice versa. Fusion, that's called fertilization. And the resulting diploid cell, just like in animals, is called a zygote. That's a fertilized egg. And then that zygote can start dividing via mitosis to produce a multicellular sporophyte. And again, this can be thousands or millions or billions or even trillions of cells, depending on the particular group. At some point in the life cycle, in the life of that sporophyte, specialized cells are going to go through reduction division, meiosis, where they go from the diploid state back down to the haploid state. And that's why we have 4 right here, because as you hopefully remember from Biology 1406, in meiosis, a single cell, in mitosis, one cell produces 2 cells, but in meiosis, reduction division, one cell yields 4 cells, 4 nuclei anyway, and that is the key feature right here. All right, a feature that unifies all of the plants is they, unlike animals, which develop an embryo where the entire animal body, and there's some variations like for animals that metamorph, they'll have 2 different versions of this, but generally the idea is there's an embryonic form of the animal where all of the structures, at least in a beginning way, are produced. All the limbs, all the mouth parts, all the tails, everything like that, wings, whatever. All of those pieces are all sculpted out early on in embryonic development, and then the animal just gets bigger and bigger. In the case of some primitive animals and the arthropods, there might be a really substantial metamorphosis from one form to another. The immature juvenile stage to the adult stage. Think caterpillar turning into butterfly. But despite the gross differences between a butterfly and a caterpillar, if you look internally at their anatomy and things like this, some key pieces are all already there. For instance, a caterpillar really does already have 6 legs. They're there, and they can be counted and identified. The mouth parts are all there, and various things like that. Plants don't do that. They do often form a small embryonic version. There's a classic embryo shape for monocot and dicot plants, the more complicated plants. But what happens is, instead of just getting larger all over, they're characterized primarily by what's called an apical meristem. So that is the tip of the root or the tip of the shoot is the growing part of the plant, and it leaves behind basically mature plant behind it. So that's the growing root and the growing stem. And then characteristic of most plants, certainly all the more sophisticated plants, is some sort of branching process where each branch gets its own apical meristem at the tip, and then that branch can keep growing. That branch of the root, that branch of the trunk, that branch of a limb, whatever it may be, the branching process carries on. And that is quite distinct. That's very, very distinct from animals. So we have this eternal growing meristematic region. A meristem is a big cluster of stem cells. You might recall that from Biology 1406. A stem cell is a cell that can divide without limit. It's not subject to telomere shortening. And so those are, effectively eternal cells, and they can continue developing forever in theory. That's what's meant by an apical meristem. So we have this cluster of stem cells that can produce all of the tissues of a mature plant behind it. In this case, we're looking at a root tip right here, and there's an apical meristem, and then a pile of sacrificial semi-dead cells right here that are produced by the meristem and constantly worn off. That's the sacrificial root trap that allows this thing to burrow its way through the soil. As it pushes, it crushes and kills its outer cells right here, but they're all sacrificial, and they're constantly being regenerated by this apical meristem here, kind of like the skin on our calluses. Very, very similar idea. But at the same time, that apical meristem produces vascular tissue, ground tissue, epidermal tissue behind it to create, all the rest of the pieces, all of the other parts of, of this organ. All right. So the early land plants were constrained because of their lack of vascular and structural tissue. Those are the non-vascular plants. So they weren't really able to get, very, very large, very tall, even though there's a fairly strong pressure to get taller, and that's competition for light. One plant, if one plant could grow over another plant, then it would shade out the other plant, whatever that was, and capture more light. So there's an immediate competition for access to light, and that's really the selective pressure to develop both vascular and structural tissues, which allow plants to get taller and bigger. And it's really the reason why we have up to 300-foot tall trees today. It's that competition for access to light. All right. So if they could get taller, they could capture more light, win in the scramble for light, and therefore do better. So, more structural complexity in their stems was a strong selective pressure. Vascular tissues are the result of that. And vascular tissues do two things at the same time. They provide structure, sort of the evolutionary forces that develop structural tissues also develop vascular tissues, and we'll find that they're highly related to each other. But those vascular tissues allow for the movement of water against the force of gravity, and the distribution of the proceeds of photosynthesis. And so that's going to be, the role of vascular tissue. And that is required if plants can get taller and taller as a result of developing structural tissue. And so we find structural and vascular tissue kind of go hand in hand in the evolution of plants. The two specific vascular tissues that you should know are xylem and phloem, which hopefully you've encountered before. Xylem is a vascular tissue composed primarily of non-living cells that passively conducts water and minerals that are dissolved in water from the soil up a shoot to the distal portions of the plant. And that's primarily the movement of water through the system is powered by evaporation and sunlight. So xylem is really composed of non-living cells, and it is a passive distribution mechanism using sunlight as a power source. Passive in the sense that there's no ATP involved in the movement of water and minerals. Phloem transport, on the other hand, is metabolically active, and it is the transport component. It's a series of living cells that are connected to each other that transport sugars and amino acids and other metabolic products of a plant throughout the entire, the distal portions of the plant. Xylem and phloem inevitably go together as vascular tissue, and they're always in close proximity to each other. Xylem really, amongst its many functions, is to provide a ready source of water for the phloem system to use. All right, so here we're looking at some cross sections and some features of plants. So plants live in a desiccating environment. So one of the things that's required for all of the plants to live on land is a means of controlling loss of water evaporation. If you take algae, all forms of algae, and put them on land, they're quickly going to dry out and desiccate and die. They're not capable of resisting that set of conditions. If they can live on land, that means they've got this, a waxy cuticle. And hopefully you're familiar with this from, personal experience. All plants, all terrestrial plants, have some measure of waxy cuticle on them. Sometimes it's very thin, very vanishing, so very hard to detect. On some plants, it's very thick and very, very obvious. But, that's just a matter of degree. This is a universal feature of terrestrial plants. So the surfaces, the green surfaces are covered in this waxy cuticle. Now, plants that produce bark, they don't produce that waxy cuticle anymore. They have a sacrificial layer of lots and lots of dead cells on the surface that's called bark. That's what bark, the outer portion of bark is. And that's very equivalent again to our calluses and our dry dead epithelium. The outer layers of our skin are really dead cells that flake off. Very analogous to bark. Plants still have to, undergo gas exchange, but gases don't diffuse through wax. Wax is not a good vehicle for diffusion of gases. So inevitably, in order to take in CO2 and to vent out oxygen, plants have stomata, which is openings that lead into a hollow, air-filled internal series of cavities called the spongy mesophyll right here. So all leaves and, to some degree, the stems and roots of plants have airways through them that allow gases to diffuse all over the place. A very similar problem that animals have to overcome. And then those stomata are regulated. That opening is regulated by a pair of cells called guard cells. Here's a micrograph right here showing two guard cells. Their state of turgor determines how open or closed this opening is right here. So this stoma right here, the single stoma, mini stomata, the single stoma is open at the moment, so gas exchange can occur. All right. Some other things. Plants, because of their exposure to increased radiation from the sun, including UV radiation, which you're normally protected from if you're a little ways below the surface of water, plants have had to produce a number of secondary pigments which absorb harmful wavelengths of light and or reflect those wavelengths of light to protect the plant tissues from, as it says right here, photodynamic damage, from what's the word I'm looking for? Ionizing radiation. There we go. Okay. Another feature of plants that we see fairly quickly, as plants became more abundant and more well-developed on land, animals started colonizing the land more and more effectively. Animals were hard-pressed to colonize land when there was no food chain to take advantage of. But as plants became more widely distributed on the surface of the planet, herbivores became much, much more abundant. And so a response, kind of derived from this idea right here, is that terrestrial plants, unlike aquatic plants, are famous for their ability to produce what are called secondary metabolites that are toxic. A lot of these are going to be alkaloids, which is to say their pH is going to be over in the the alkaline region, pHs of 8, 9, and 10. So to us, those things taste very, very bitter. And almost universally, they're going to be toxic or poisonous to some organism. All right, and so that is a really, really common characteristic of terrestrial plants, particularly the more sophisticated and robust terrestrial plants. We don't see so much of those secondary metabolites over here in the bryophytes, but by the time we get to the lycophytes and certainly the pterophytes, ferns and their allies, we have lots and lots of secondary metabolites, which make these things increasingly challenging for animals to eat. And so, of course, the way evolution works is those animals that do develop biochemical or structural modifications that make them tolerant of these secondary metabolites, then those animals can continue eating these things and being herbivores. And that's what we've really got on land, is kind of an evolutionary arms race between the defenses of plants against being eaten and the adaptations of animals that allow them to continue eating plants. And that's very characteristic of the evolutionary process. All right, so that's a little bit about poisonous secondary metabolites. Nicotine in tobacco plants is an example of one of these things. The selenium in potatoes and tomatoes and various other plants that are toxic to a number of different animals. That's what those substances are. Surprisingly, the materials that we're attracted to in chocolate and coffee are anti-insect herbivore compounds meant to keep insects from eating those plants. Those metabolites have interesting effects on our hormonal regulation of our central nervous system and our endocrine systems, and we've developed kind of a symbiotic relationship with a number of those different compounds, since our love of coffee and chocolate and various other psychoactive compounds. A lot of other animals have done that as well. And then that kind of leads to this last idea right here, is that plants have co-evolved with a lot of animals by manipulating them, both with these secondary metabolites, which oftentimes can change their behavior. That's what we mean by a psychoactive compound, and these straight-out rewards. One of the things plants produce that they can always share in exchange for some service is sugar. Since they are primary producers of sugars, all other animals are looking either directly or indirectly for sugars and other carbon-based metabolites like amino acids and lipids that they can use as food. And so plants have produced these substances to co-opt the behavior of animals and primarily aiding them in reproduction. And so that is directly through pollination, fertilization of eggs by sperm in the pollination of flowers process, or by dispersing their young, by taking their seeds either internally or externally some great distance from the parent and aiding in the dispersing of the young. So, a relationship that was initially antagonistic between animals and plants right here increasingly becomes symbiotic as we move forward through the development of more and more sophisticated plants. So going back to this image right here, these things are just generally eaten by various animals, and their only real defense is to outproduce them. But by the time we get to the lycophytes and certainly the pteridophytes, they've developed quite a few structural features that make them harder to eat, and they produce secondary compounds that make them more toxic to eat. And in some cases, the secondary compounds alter the behavior of the animals in a way that's beneficial to the plant. Both of those processes, structural adaptations that make them harder to eat and chemical compounds that alter the behavior of the herbivorous animal or other animals, are increasingly seen in the gymnosperms, pines, cycads, junipers, and all of those guys, and then taken to a terrific level of sophistication in the flowering plants, which many flowering plants have permanent, dedicated symbiotic relationships with one or more other species of animal that increase the ability of these things to reproduce and live. And that's not to mention the relationship between all of these things in fungi and all of these things in soil bacteria. There are symbiotic relationships between all terrestrial plants and soil-based fungi and soil-based bacteria. All right. Oh, another example of this again. So a new term we'll put up in here. Vascular plants are also oftentimes referred to, so that's all of these guys right here, as tracheophytes. And that is because they have these cells called tracheids. So a little bit about that term, tracheids or tracheophytes. So here's an image just pulled from YouTube right here of a human tracheal and bronchial tree, for the respiratory system leading into the lungs. And the trachea is characterized by these rings of cartilage in between layers of connective tissue, and down here in the terminal bronchioles, even some muscle tissue. That's a trachea right there. So when we're looking at tracheids, well, this didn't come out very well, but this picture right here, let me see if I can zoom in on this. These are tracheids in plants, and there is a vague similarity between them and the trachea, which is why people researching these use that term. There's a passing similarity in the tracheids right here, in that they have rings of reinforcing structure holding the cells open against a suction, so that they can conduct the fluid that's moving through them, even if they're under some sort of suction, which is why our trachea have these rings of cartilage, so that when we breathe in, the suction of breathing in doesn't cause these tubes to collapse like they would if they were unreinforced. So tracheids have that same, they have a structural feature that makes them strong and resist collapsing. So here's some electron micrographs of tracheids right here, various forms of tracheids. These are some right here, some vascular tissue running through a plant. Here's some tracheids right here. Here's some great isolated tracheids. And, the key thing is they've got a much thicker cell wall right here, and it's oftentimes in kind of a repeating ring-like structure, kind of like a trachea. So that's where that term comes from, and there are some real similarities. So let's go ahead and get rid of that. And so tracheophytes. All right. So now let's work our way through these organisms and take a closer look at them. And in this PowerPoint, we'll restrict ourselves to the seedless plants. So the streptophytes, is a large group of green algae that are the parents of all of the terrestrial plants. It's a particular group of green algae that includes This particular group called charophytes, and we'll take a look at a picture in a minute, you are needing to know these guys, their identity, their name, and their structure. Charophytes. That is a group of multicellular, colonial, so multicellular but not obligately multicellular aquatic photosynthetic organisms. They have the same chlorophylls A and B that terrestrial plants have. They share the same carotenoids, which are blue light absorbing pigments that appear red to the naked eye right here. And unlike all other types of algae, they store carbohydrates in a polymer in the form of starch. So that's a unique characteristic of these things called streptophytes. The rest of the green algae do not share those characteristics, and these are some that you might or might not be familiar with. Sea lettuce is something you find on rocky shorelines on temperate coasts around the world. Spirogyra and volvox are freshwater filamentous, in these cases, and spherical algae, very, very small. All right, so here's a charophyte right here, which, as the slide says, this is Chara right here. It is a invasive, noxious weed in Florida. It comes from Asia. There's things that eat it in Asia that are not present in the waterways in southern Florida, and so it accumulates in huge amounts and kind of takes over those waterways. This is an algae. It's not a plant. It doesn't have a cuticle. It doesn't have any vascular tissue. It doesn't have any really serious structural tissue, but it does show some of the characteristics of plants. It has an apical meristem, so it continues growing from one bushy end right here, leaving behind the node, internode, internode, and the potential for branching that occurs at each one of those nodes that's characteristic of plants. So that's a real structural feature of plants. This thing gets longer and longer, and it grows from the tip. It's the tip that's growing. These bits aren't growing right here, but they're left behind and continue functioning. All right. So now let's talk about bryophytes, which is, that very large group that includes the mosses that y'all are most familiar with. All right. There are 3 phyla that we need to know. The Hepatocophyta, liverworts. There should be a W right there. I'm sorry. I'll see if I can fix that later. The Anthocerotophyta, Anthoceroto, I never say that, so pardon practicing it. Those are the hornworts. And then finally, the bryophytes themselves, the guys who give the name to this larger group right here, the mosses, the true mosses right here. All right. And as you can see, there's about 25,000 species, so they're fairly abundant. And even though you can find them in deserts, and they really do live in deserts, so we'll find bryophytes all over the planet, but never in really, really large concentrations unless we get in sort of ideal damp habitats, and that's where they do the best when we find the most of them. All right. As a rule, they lack lignin, which is a reinforcing protein that you find in the cell walls of more sophisticated terrestrially adapted plants. And because of that, amongst other things, they lack any true vascular tissue. They've got some specialized cells that allow for the conducting of both water and metabolites, and that's simply that they have cells that are cross-connected to each other by lots and lots of plasmodesmata, so that they can share cytoplasm. But that's the limit of their specialization, is that, there are many cells through the bodies of the bryophytes that are cross-connected to each other to share cytoplasm. But otherwise, they're normal cells with all of the usual organelles and central vacuoles and other features internally. All right. When you are looking at moss, you're looking at a gametophyte, which means it is haploid. All right. The sporophytes, you have to know what you're looking for to find them. They're very, very hard to detect. And importantly, they're metabolically and structurally supported by and dependent on the gametophyte. So you can think of the sporophyte generation as almost being a parasite of the gametophyte. That's characteristic of all of the bryophytes. All right. So the gametophyte is dominant. There is a reproductive structure, a female reproductive structure, a multicellular structure that produces a single egg that is called an archegonia. Please know that term. The male equivalent is called an antheridia, and you may recognize this term from discussion of flowers, the male parts of flowers. And the antheridia produce sperm, and that's plural, so they can produce lots and lots of sperm cells. In non-vascular plants, the sperm are flagellated, just like in animals, and they have to swim through water to get to the egg. All right. So when that happens, we fertilize the egg, and the egg is in this archegonium, and it's going to develop by, it's going to germinate, which is to say it divides via mitosis to produce a diploid sporophyte. And that sporophyte is going to be nutritionally dependent on the gametophyte and living in the gametophyte. Eventually, that gametophyte, it's going to have a structure, not an archegonium, not an antheridium, but instead a sporangium, and it will produce spores via meiosis, haploid spores that grow into a fresh generation of gametophytes. So here we're looking at the liverwort right here, and this flat leaf-like structure, for the record, is called a thallus. All right. This is pretty characteristic of these things right here, and this is all gametophyte tissue right here. These are all gametophytes, haploid tissues. All right. All right. And if we look kind of closely at them, what we'll see is that somewhere on the thallus of the liverwort, we're going to have and classically they have gender, so there's a female or a male in a version of this. They produce a structure on their undersides that reaches up, an antheridial head right here, and it produces a whole bunch of these antheridia, which is a multicellular thing that produces sperm via mitosis. And so these are like little capsules full of sperm. Eventually they'll burst open, and the flagellated sperm cell, here it is with its two flagella, will swim through a layer of liquid, like a drop of water or a film of water over all of these. So classically they reproduce during the rain or when they're underwater temporarily. And the sperm will swim, and they're attracted by a chemical trail to swim directionally towards an archegonium on a female portion of a liverwort or a female plant if they are sexually dimorphic, and fertilize that egg. And once that egg is fertilized, so here's an archegonium and here's that egg, the sperm, from these antheridia, swims into that archegonium right here. There's a little pathway right here, reaches the egg, fertilizes the egg, and then the egg starts dividing via mitosis to produce an embryo. And eventually the sporophyte becomes this large, mature, multicellular thing still living on the gametophyte. The green is gametophyte, gold right here is sporophyte. And this diploid sporophyte, when it's mature, it will have cells that divide via meiosis, reduction division, meiosis, producing haploid cells right here. And those haploid cells are released classically during dry weather. Sometimes it's wet, it just depends on the particular species, but oftentimes during dry weather, so the spores are capable of blowing away on the wind, and when they germinate in some new location, a new gametophyte can grow in that location. So that's their means of dispersal. Reproduction is via sperm and eggs that occurs locally. Dispersal to new locations is via spores, which are desiccation resistant. The spores are covered in a tough protein called sporopollen, and that word is not an accident. We reuse the second part of the word, not spore, but the pollen, as the description of the covering of the gametophytes in flowering plants. So we'll see that word again, sporopollen. Remember, they're connected to each other. So that's the liverwort, and that's typical. The life cycle is typical of all of the bryophytes, but the details here are for the liverwort. Here's an example of a hornwort, and as you can see, the reason it's called a hornwort is because it has these flat, sort of lobate leaves like the liverwort right here, but it has these tall structures reaching up, and these are the sporophytes, and these are the spore capsules, the sporangia on the surface right here in orange. Those are the sporophytes, and they are dependent on the gametophytes down here, the green things, for their nutrients. And their reproduction is going to be really similar. The details are essentially the same as with the hepatocyte, the liverworts right here. Now the bryophytes, again, they have the same reproductive structure. The details, the major details are the same. The tiny details are different. So a big difference between the liverworts, hornworts, and the bryophyta, the mosses, is that they look more like a plant in the sense that they have an elongated stalk right here, with lots and lots of pseudophylls, which is false leaves sticking off of them. There's no vascular tissue in here. There's no vascular tissue in the leaves. So it's just, you know, a big multicellular mass. But it's been modified to create more surface area for more photosynthetic activity. And they are nominally bigger than these things or these things. Sorry, here. They're, you know, most mosses can be quite a bit larger than a liverwort or a hornwort. The mosses can really be quite large. Here's some examples of mosses right here. And, you know, kind of like the hornworts, we're looking at the gametophytes down here, green photosynthetic haploid, and sticking out of here as parasitic dependent structures are the sporophyte generations. Spore capsules out on the tips, we're looking at them in detail right here. And those spore capsules are going to burst open and release spores, oftentimes under dry conditions so the spores can be blown far away, sometimes in wet conditions so they can be washed away. But the spore is the dispersing agent. All right, so we're looking at the life cycle right here, kind of in cartoon form right here. So a spore capsule bursts open. One way or another, a spore is released. The spore germinates and starts growing into a fresh little gametophyte, you know, small at the start, and continues growing. The gametophytes spread vegetatively. They have a horizontal runner called a rhizoid, and vertical structures called pseudostalks. You know, these are the gametophytes, the non-vascular stems. And inevitably, they will produce, you know, reproductive structures, and those are archegonia to produce eggs. One archegonium produces one egg. Antheridia, one antheridium can produce thousands of sperm cells. And at key moments, usually in association with water, the sperm are released. They must swim to hopefully another plant so that we can have, you know, outcrossing and reproduction, and swim into an archegonium. They're attracted by a chemical cue and work their way down into that archegonia and fertilize an egg. That egg, now the zygote will start dividing via mitosis to produce the dependent sporophyte growing up out of the gametophyte, kind of as a nutritionally dependent parasitic relationship right here. And then we come all the way around full cycle here. This is just another illustration of the same thing done, looking at minium, which is a classic example for a traditional sort of example-based biology laboratories. You would look at slides of this organism and look at all these reproductive structures here. All right, and so here's some photographs of it again. So when you're looking at moss, you're looking at a gametophyte. If it's the right time, if their life cycle, you'll see emergent sporophytes right here. And here's a close-up of the sporophytes and their spore capsules. At some key moment, those things will dry out, pop open, and release a bunch of spores. Spores are extremely desiccation resistant and very, very long-lasting. And that is a consistent characteristic. A spore is able to last over huge amounts of time. We have spores routinely that we find now, especially in permafrost that's melting in the northern hemisphere, spores that have been frozen for thousands of years that are viable and germinate again. So that is a great way for them to transcend time and space. All right. The big innovation is the development of vascular tissue, which also provides for mechanical strength and structure at the same time. So the development of vascular and structure tissue is kind of a simultaneous event in terrestrial plants. And the first cells that we see are the tracheids, these specialized water-conducting cells that, and I'll take you back to this image right here. Here's a cross-section of some tracheids, and they have a very, very thick, tough, oftentimes in more developed plants, lignified cell wall right here. And that tough cell wall gives the plant structure. So the stem is able to get much taller and much tougher, be able to resist the force of gravity and get quite a bit taller. So tracheophytes have these tracheids, which give them structure, which allow them to get quite a bit taller and kind of compete effectively for light. And the tracheids allow for the rapid movement of water from the absorption structures, whatever's plugged into the ground, nominally the roots, all the way up through the stem to the distal portions of the plants. So that's what we're seeing when we see the appearance of tracheids in particular. Xylem followed more distantly by phloem. Now, this may seem a little bit counterintuitive right here. Xylem tissue is lignified for mechanical strength, and it is impermeable to water. The purpose of xylem tissue is not to use water or immediately transmit water, but to conduct water over great distances. And so that allows us to, build a taller plant, grow a taller plant that can, get higher up, have more access to light, and in the wind, their spores are able to be blown more effectively because they're higher up, so they fall further, and if they can pick up a stronger breeze, they can be blown further. That primitive, let's go back and find it. These water and nutrients that are circulated inside specialized conducting cells, those are the ancestors of what will become phloem tissue. All right, so here we're talking about phloem right here. Phloem transports sugars, proteins, and other solutes that consist of these specialized cells called sieve elements right here, and then some supportive cells that keep them alive. They are derived from those just basically cross-connected cells in mosses and the rest of the bryophytes. But the difference is these cells have become highly specialized for the movement of cytoplasm from cell to cell to cell. And they've given up a lot of their internal structure, and that is taken over by these supportive cells, which aren't structural support, they're metabolic support. That we have these called companion cells or support cells that keep the sieve cells alive. And that's phloem. Phloem is a combination of these sieve cells, which are the conducting cells, and then these metabolic support cells that keep the sieve cells alive. Kind of an interesting feature. And so plants that contain these structures are called tracheophytes. All right, so when we get conducting tissues, when we have vascular tissues, that's the point of origin for a true stem and a true root. So the bryophytes have these rhizoids, which are just extensions, of their stem that run across the surface of a substrate, but they don't really burrow down into the substrate. So if you've ever looked critically at a moss, if you've looked critically at a moss and explored them and tried to manipulate them, you'll find that mosses peel away from their surface quite easily. They never have roots. They may glue themselves onto a surface. Mosses growing on rocks or bark are oftentimes intertwined with that surface as kind of a mechanical adhesion to the surface. But they don't grow into the surface. They don't have any penetrating structures. That requires both structure and vascular tissue. So the tracheophytes are the first plants that actually have roots that can, penetrate into soil and make contact with resources that are buried deeper in that soil. All right, so those true, roots appear later on, as it says right here, not in the bryophytes, but in the more derived, where is it? Oh, the image. These groups right here, we see, vascular tissue in the lycophytes, then pteridophytes, and so on and so forth. All right, and so that's the origin of roots. So again, these guys right here, here's some moss that's been pulled off of its substrate, and there aren't any roots. The rhizoids, which is just the horizontal, where was a picture of it earlier? Here's the rhizoids right here. Provide some mechanical adhesion, but that's about it. They're not really absorptive structures, and they don't penetrate deep in. All right. When we do see true roots, that's where we see the establishment of these symbiotic relationships with mycorrhizal fungi in soils. And so those mycorrhizal fungi increase the absorbing ability of the roots, as it says right here, particularly for phosphorus through proton exchange. And the plants share sugars with the fungi in exchange for the fungi reaching out for mineral nutrients in soil. So that's a symbiotic relationship right there. And then nitrogen-fixing bacteria take N2 gas out of the atmosphere and convert it into ammonia, NH3 or NH4, or nitrous oxide or nitrite or nitrate into NO2 or NO3, all of which dissolve in water and those can be absorbed by the roots of terrestrial plants. So all terrestrial plants have a symbiotic relationship with nitrogen-fixing bacteria as well. And in the case of the legumes, a group of very, very derived plants, they actually grow nitrogen-fixing bacteria inside specialized organs called root nodules. But that's for a little bit later. All right. Leaves, sporophylls, and strobili right here. So a leaf is a increased surface area structure coming off of a stem that allows for more surface area for photosynthetic activity. That's the function of a leaf. And so the bryophytes have what are called false leaves in the sense that they don't have any vascular tissue. And so the seedless vascular plants have developed those, true leaves, which have vascular tissue in them. All right. So some terminology: a microphyll, fill right here is in chlorophyll, is a very, very small leaf, with maybe one vein running through it. And then the megaphylls are larger, more complicated leaves that have branching patterns of venous structure in them, and so this allows them to get quite a bit larger. Sporophylls, as it says right here, are leaves that are modified to hold sporangia, and so that's like the structure on the underside of a fern frond, a cone on a cone-bearing plant, or a flower on a flowering plant. Those are all modified leaves that have been modified to do this sort of function right here. So leaves and the stems that they're attached to get modified to make reproductive structures. Sporophylls generally, and strobili, as it says right here, are particularly those cone-like structures that we see on pine cones. Those are all modified leaves. These are all versions of this idea of a leaf, which is a thing that branches off of a stem and provides reproductive function. All right, so when we have vascular plants, but still seedless vascular plants, one of the big changes we see is that we switch from the haploid being the dominant life form to the diploid organism being the dominant life form. All right. Now, one thing that they share very, very strongly with their bryophyte ancestors is the dependence on water for fertilization. So even the vascular plants still have antheridia that produce open, free, swimming sperm cells that must swim to an egg and make contact with that egg and fertilize it. So reproduction, at least, requires lots and lots of water available, either a layer of water over the reproductive structures or being temporarily underwater. So that's why we still find associate ferns with sort of damp, moist, protected foresty sort of regions. Because they need that for reproduction. Now, there's plenty of these organisms that can live in dry, harsh parts of the world, but they have to get wet in order to reproduce. All right. So the sporophyte is now the dominant organism. The gametophyte becomes reduced, smaller and smaller and smaller, but we still have that flagellated sperm that has to swim to an egg cell. All right, let's take a look at the Lycopodiophyta, and so lycopods. These are club mosses, and so these are very, they look superficially like mosses, but much larger, much bigger. And if you want to visualize, what the early ancestors of dinosaurs and some of the, first big terrestrial vertebrates, herbivorous vertebrates, were eating, it would have been things like this. Club mosses would have been early dinosaur food, herbivorous dinosaur food. And as you can see, we've got 3 different major groups right here: the club mosses, the spike mosses, and the quillworts. They're all members of this Lycopodiophyta group. All right. None of them are real mosses. They all are diploid dominant, and they all have vascular tissue. And as you see right here, because they have vascular tissue, the sporophytes have roots, and those roots are going to be associated with soil mycorrhiza, which allows them to get much more out of the soil in the way way of nutrients, get a heck of a lot bigger, and do just quite a bit better on land. So, you know, the vascular tissue made these things much, much more successful on land and be able to be the food basis for a much more complex terrestrial ecology, including large vertebrates. So here we're looking at some small club mosses. These still exist today, but you know there are extinct versions that were really, really huge in comparison. And the sporangia are grouped together, so a whole bunch of a bunch of stem is modified with really closely packed little modified leaves that each have a sporangium on them, and those are grouped together into this structure called a strobili. So it's a little hard to recognize the relationship right here, but these have a relationship to pine cones. It is a kind of evolutionarily, this is a primitive version of the pine cone idea. Here's some equisetum. As you can see right here, it's really a marsh-loving organism, although there's some more dry weather versions. And notice these things structurally look more than passingly like, we'll go all the way back to the beginning of the presentation, this. There's more than a passing similarity to these things. This is a purely aquatic organism. These guys are terrestrial organisms. Let me get back to them. But you know, structurally, there's a lot of shared genetic control for the way they grow. The difference is they've got structural and vascular tissue in these stems right here, which allows them to grow vertically against the force of gravity, and they have a waxy cuticle on their surfaces that allows them to resist desiccation. They have stomata, those openings protected by guard cells that allow them to control their exposure to the atmosphere. So they open up their stomata to exchange gases. They close their stomata to protect against water loss when they're not in a hyperabundant water environment like this. And here are the horsetails right here. Again, there's more than a passing similarity structurally to Chara, to the charophytes, but they've taken this to a much greater degree, and it's a much more robust structure right here. Lots and lots of structural / vascular tissue in here, and roots. An interesting bit of information right here. Horsetails, it says right here, apparently were used as scrubbing brushes, and the key thing is they've got silica. They have nodules of silica in the epidermal cells covering their surfaces right here, which makes them really tough and abrasive. And so that's why they might have been used for this function. The purpose of the silica in their epidermis is to keep things, to dull the mouth parts of chewing organisms. So things that would try and eat these things would be wearing down whatever they're chewing and biting with because of the silica right here. So that is an anti-herbivory adaptation. And these are the whisk ferns right here, and they're strobili at the tips right here. You know, are very tiny and spherical right here, so they've got that knob-shaped little sporangium right here. But these are the places where they're going to be producing spores. They'll be producing spores right here, and this whole thing is a sporophyte now, not the gametophyte, but it is the sporophyte. This is a sporophyte. This is a sporophyte. And then finally the ferns themselves, and that's the one where we'll take a look at their life cycle in some detail. These are the most abundant of the seedless vascular plants, as it says right here, about 20,000 species, ranging, you know, all over the world right here. And hopefully most of y'all are somewhat familiar with a fern. And so again, when you're looking at a fern, you're looking at a sporophyte now, not a gametophyte like when you're looking at mosses, at bryophytes. There is the classic fiddlehead, which is an easy way of knowing that you're looking at a fern. If you can find a young developing frond, it should have this curled shape right here, which I believe is universal to the group, these fiddleheads right here. And, you know, that is a decorative motif on string instruments, hence the name fiddlehead. All right, so here's the life cycle of these guys. We still have Archegonia, a multicellular structure producing a single egg. Antheridia, a multicellular structure, and these are on gametophytes, producing sperm cells. The sperm cell is flagellated, if you can see these little lines right here, and has to swim through a film of water to an egg on another plant for sexual reproduction to occur. The fertilized egg then starts, you know, germinates inside this archegonium right here, but unlike in the gametophytes, sorry, gametophytes. Unlike in the bryophytes, mosses, and all of their relations, the sporophyte becomes self-sufficient. It actually grows out of the gametophyte and destroys the gametophyte and grows into a very big root, producing, stem producing, um, frond producing thing called a fern plant. And when it's reproductively mature, on the underside of the fronds, and the patterns vary from species to species, but you'll have these multicellular structures right here called sori, and each of those sori has a bunch of sporangia under it, and that would be equivalent to these sporangia right here on the Psilotum, on the whisk ferns, or these strobili right here, where is it, on these club mosses right here. Same sort of idea, just different arrangement, same function. So this is a multicellular structure on the underside of the frond right here that produces spores via meiosis. Those haploid cells are the means of dispersing the animal to lots of new locations. Spores can last thousands of years, blow in the wind for hundreds or even thousands of miles. And when they land somewhere, they can germinate into a new gametophyte, which is a very small thing. You'll be hard-pressed to ever find this in the forest. You'll see lots of ferns, but you've got to know what you're looking for to find the little tiny sporophyte somewhere on the ground. It doesn't live very long. It doesn't have any roots. It doesn't have any big stem. All it does is produces eggs and sperm to complete the life cycle and get back to the diploid stage. And that's characteristic of all of our vascular seedless plants, is that the gametophyte is now very reduced as, a short-lived sort of specialty plant. It is photosynthetic and green, as you can see right here, and multicellular, but it only lives long enough to produce gametes, a male and a female version of this. Sometimes they're hermaphroditic. They'll be male and then female at a different point in time. They don't fertilize themselves, though. There's got to be another one somewhere nearby where the sperm can get to for fertilizing an egg on the other one. And we complete the life cycle and get back to the dominant thing that can live for years and years and years and get really quite large, the mature sporophyte. So here's an example of sori, which just have lots and lots of sporangia in them. Each of these is a big structure with a whole bunch of spore-producing structures within it. And this is the this is an error, right? Oh yeah, here's a new sporophyte, a new fern growing out of the gametophyte right here. So if we look back at this life cycle picture, that sporophyte right here is growing from this fertilized egg in an archegonium on a gametophyte. And in time, the sporophyte will grow into this great big thing, and this will be gone and dead. The gametophyte will not last long. Just a little bit of a trivia bit, a very tiny plant that is a fern called a zola is an aquatic organism. It lives on the surface of ponds, freshwater aquatic systems right here, and has nitrogen-fixing bacteria, cyanobacteria, photosynthetic bacteria, living in it. So, a zola is a really, really tiny fern, lives on the water, not in the water, but on the water, on the surface, and harbors these nitrogen-fixing cyanobacteria that produce soluble nitrogen, which all living things need in order to make their nitrogenous bases for DNA and RNA, and the amine groups for amino acids and proteins. No nitrogen, no life. And N2 in the atmosphere, plants can't get a hold of it all. They have no means of grabbing it. Only nitrogen-fixing bacteria can take N2 and turn it into ammonia or nitrate or nitrite. All right. So some ecological roles of interest, when we look at ferns, because they have roots and they secrete chemicals, in their roots, including acids, they're capable of breaking down rock pretty substantially, forming soil, and, contributing to, a substantial change in the terrestrial environment. Soil is created by, by erosion, chemical and mechanical erosion, but that is amplified terrifically by the activity of plants. And then retention of that soil is all about plants. All right, I think that is it. Yeah, so that is it for this particular lecture, and the next lecture will be on seed plants.